Embark on a captivating exploration deep into your DNA, the blueprint of your life. Join us in this video as we unveil the hidden narratives embedded in your genes through the lens of genetic testing. Get ready to delve into the intriguing convergence of science, identity, and the future of personalized healthcare. First, let's discuss what the table of contents of this video will be. First, we'll be looking at what is a genetic test, what are some factors associated with genetic tests, what are the different types of genetic tests, what are some criteria used to recommend an individual, looking at some information from an expert in the field, as well as discussing what is consensus. First, let's discuss what is a genetic test. A genetic test involves analyzing and studying DNA by evaluating an individual's genes and chromosome and determining if the individual has a specific condition. Did you know, as of now, we have 77,000 tests that are being utilized globally? What are some factors associated with genetic tests? Your genes, chromosomes, and proteins are all examined during genetic testing. By evaluating the genes, we are looking at the DNA sequences to see if we can find any mutations that may cause genetic disorders. These tests can include looking at the whole nucleotide, the DNA building block, the whole genome, which is all the DNA in an individual, or multiple genes. Chromosomal genetic tests include evaluating pieces of a chromosome, finding an extra copy, or looking at the whole chromosome to identify any causes of a condition. Then, protein tests look into your enzyme and protein activity, and if any of the numbers are out of ordinary, this can indicate a cause for a genetic disorder. Now let's talk about what the different types of genetic tests that are there. This can include molecular tests, chromosomal tests, gene expression tests, and biochemical tests. Molecular tests look into changes in the DNA, and this can be tested through a targeted single variant, which looks at a specific variant that can be found in a gene. It is a specific variant that is identified that causes the genetic condition. For example, those with sickle cell anemia have a mutation in their HBB gene that causes this disorder. This test is most commonly used on family members where a specific variant has already been identified and they are testing to see if it is present in the other member. Other molecular tests can include a single gene, which looks at one gene panel, which looks at multiple genes, and whole exome sequencing, which looks at the majority of an individual's DNA. Another type of test is chromosomal tests. As mentioned before, they analyze pieces or the whole chromosome. If, for example, an extra copy of a chromosome is found or is missing, this is referred to as trisomy for extra and monosomy as one chromosome. Other changes can include deleted or rearranged pieces of the chromosome. Thus, different changes to the chromosomes can be identified to pinpoint a genetic condition. For example, if there is an extra copy of chromosome 21, that leads to Down syndrome. Another type of test is gene expression test, which evaluate which genes are expressed and not expressed, on and off. These tests observe the mRNA found in cells to analyze their expression. Having too much or little expression can be indicative of a genetic condition. Lastly, as mentioned before, biogeochemical tests analyze the enzyme and protein activity, and if any numbers are out of the ordinary, this can cause a genetic disorder. What are some criteria used to recommend an individual? Doctors may advise an individual to undergo genetic testing based on family history and history of cancer in the family. Ovarian and breast cancer, for example, are seen to run in families. It is important to note, however, that genetic testing only provides information about the disease as a whole. Other genetic factors such as your diet, where you live, how you live your life, can contribute to genetic conditions. It is crucial to share your family history with doctors. This information can uncover inherited genetic conditions impacting family members, assess the risk of developing these genetic conditions, and guide recommendations for genetic testing.
Hi, my name is LandryNfonsam, and I'm a clinical laboratory scientist in genetics at Hamilton Health Sciences and St. Joseph's Healthcare Hamilton. I'm also an assistant professor in the Department of Pathology and Molecular Medicine at McMaster University. Now, there are many benefits to performing genetic testing, most of which have had tremendous positive impacts in our lives. Now, the first benefit that I'm going to address of genetic testing is the early detection of disease-causing mutations can lead to early intervention. Now, in the context of prenatal testing, It provides couples with informed choice on how to manage the risk of having an affected child, including making decisions about whether or not to keep a pregnancy. It allows physicians to plan prenatal treatment of fetuses with a genetic disorder or birth defect. For cases where prenatal treatment is not available, it alerts parents and physicians to arrange for appropriate management for the impending birth of an affected child. This may include psychological preparation of family members, pregnancy and delivery management, and postnatal care. In the context of carrier testing, it helps identify individuals at risk of a genetic disorder. For example, early detection of BRCA1 or BRCA2 mutation that causes breast and ovarian cancer permits individuals to make the choice of initiating prophylactic measures like mastectomy and or salpingo-oophorectomy. Predictive testing for HTT mutations that cause Huntington's disease allows individuals who are carriers to potentially make appropriate plans for the future, like choice of job, lifestyle, plans for later in life care. For pre-implantation genetic testing, it offers carriers of pathogenic variants who are scared to pass on the mutation to their offspring a way out to be able to have healthy children. Genetic testing can be performed on embryos during IVF and only healthy embryos selected for implantation. For biochemical tests, early intervention can be critical, especially for cases where decompensation episode can be very acute and can rapidly lead to death. For example, elevated levels of phenylalanine in phenylketonuria or elevated levels of ammonia in ornithine transcarbamylase deficiency, which are both molecules that can cause brain damage. Now, there are limitations associated with genetic testing. Genetic testing does not always give you all the answers. There are limitations. For example, genetic testing can be limited by the testing methodology. A negative result does not rule out the presence of a pathogenic target region. Also, mutations might be present in a difficult to sequence region and therefore might be missed. Another limitation is that for some single analyte tests, only one sample can be analyzed.
What is consensus? In this context, consensus refers to securing approval for the genetic test, known as informed consent. This involves providing individuals with sufficient information to understand the test and underscore its voluntary nature. Only adults capable of making medical decisions can agree to undergo the test, while for individuals or children unable to make the decisions, a parent and or legal guardian with decision-making authority can provide consent on their behalf. Consent is usually acquired through a doctor and or genetic counselor, and once they agree, they are required to sign a consent form that includes information such as the type of the test, how the test will be completed, such as a blood test, what the results may mean, the possibility that the results may reveal any family history, and then require a signature. If you are interested in more videos like this, please subscribe to our YouTube channel, Demystifying Research. Thank you.